All right, welcome back. I hope you're having an amazing time so far. Our next speaker is Kevin Brown. He is an Associate Director of Data Science at Bristol Myers Squibb. Please join me in welcoming Kevin to our virtual stage.
It's great to be here. I'm going to talk to you on behalf of Bristol Myers Squibb, a large pharmaceutical company, about computer vision in drug development, where we try to leverage the power of multi-modal data and take computer vision just from something looking at merely images to integrating with the patient as a whole. And when coming to Bristol Myers Squibb and being asked to head up their imaging for medical AI, the first thing that we asked ourselves, well, what will always be true? In this case, we knew that always you will have a use case that drives any computer vision application in industry, whether that's for radiologists using artificial intelligence software to make diagnoses or recommendations as clinicians, whether that's computer vision applications in self-driving cars or manufacturing, or whether that's something in order to help a clinician work with a patient or a patient data set. Always at the foremost, we will have some sort of goal that we're trying to achieve. At the same time, that will interface with two other things. One is the data that we have available, and the final is the algorithms we use to link that use case and data. And coming from a mathematical background, the first thing I thought, well, this is nothing more than creating a feature set X and asking X how it relates to our use case using algorithms as some sort of leveraging point. So knowing what will always be true in terms of a feature set and relating that to a use case through algorithms, we can take a step forward in terms of what is true today. Well, in medical imaging, what's true is that we use medical imaging more and more on a day-to-day basis in order to inform clinical decisions. But more than that, we also use it in order to inform pharmaceutical decision-making points. In clinical trials, medical imaging is the #1 non-overall survival endpoint in oncology studies. So every single lung cancer trial that you see, for instance, for metastatic non-small cell lung cancer, is going to have an imaging component. And in that imaging component, radiologists are going to measure particular tumors, and they're going to relate that to which drug you're on and other relevant factors and variables. But the other thing about this use case is that we know that clinicians are not perfect. The data that we have available, we know that in late stage drug development, imaging data is extraordinarily rich. It gives information not only in terms of the phenotypes of the tumor expression, but also in terms of other prognostic factors such as sarcopenia or other imaging factors that might indicate how long a patient has to live or whether they would respond to a particular kind of treatment. We also know that labeled data is carefully curated and managed. And this is something people outside of the pharmaceutical industry don't necessarily appreciate, where data can be much more noisy. But in a clinical trial setting, that data on a regulatory basis is very carefully curated and managed and gone through multiple steps of quality assurance. And not only that, it's paired and it's linked across different patient variables or attributes. So when we speak about computer vision in the pharmaceutical industry, we're not merely speaking about pixels or three-dimensional voxels. We're speaking about pairing it with clinical data, such as sex, how the patient is feeling on the particular day of the screening, body mass index, whether they have a history of smoking, various histopathology images that are also analyzed and curated. And those get paired with relevant outcomes that are outside the realm of something you can get from a data set like ImageNet or any of the many other off-the-shelf academic data sets. In this case, we can pair it with OS, as we call it, or overall survival. PFS, progression-free survival, how long a patient in rough terms survived without their tumors growing in certain ways or new tumors appearing in solid state tumors, or AEs, adverse events. And it's this pairing that takes us from computer vision into the realm of multimodal data. And what's particularly amazing about the data sets that the pharmaceutical industry has access to as a data generating machine in terms of drug development, these millions of images that are collected are labeled. They get labeled rigorously both by investigators at individual sites where clinical trials are carried out. They also get labeled at core labs where we have very specialized radiologists who do this for a living, who are very, very good at labeling images in terms of the size, the shape, or other aspects of the appearance of, for instance, solid tumors in non-small cell lung cancer. And pairing this with the clinical data, we can start to ask, what are the algorithms that would allow us to bridge the use case of predicting overall survival, or progression-free survival, or choosing which patients should go in which treatment arm, or making predictions about how they would respond to treatment. And we can ask ourselves what algorithms lend themselves in a natural, unforced way to the multimodal integration of this paired, curated, integrated data sets that we have access to. So we know today that state-of-the-art methods are readily adapted to clinical images. You can right now go to, say, Papers with Code. You can pull the latest algorithms, say, from Microsoft Research, a shifting window transformer, for instance, for object detection and segmentation, or perhaps image classification. You can apply these to your images in almost a one-to-one correspondence. Sometimes you need to shift for a 3D nature of medical images. Sometimes you don't. And we know that these algorithms are typically public domain. So one of the amazing things about the last 10 years is the academic and industry partnerships have produced a plethora of algorithms that are ready for use, including pre-trained weights that allow you in a matter of hours to do what used to take a matter of months or years in order to develop, test, and validate. So when we ask ourselves about what's true today, how can algorithms be used to bridge the data that we have access to and the use cases, the implications are sort of threefold, fourfold, and very clear, and all related, which is good news because it tells us we can use a similar approach. And there's a vast amount of horizontal scalability across the different applications that we might use, which we'll touch on in a minute. So the obvious thing is that with millions of labeled images, you can build more accurate algorithms. Time and time again, we know that data is king when it comes to machine learning. If you take one algorithm and you add more data, you will typically, depending on the quality of that data, do much better without making any other architectural changes. What you can do with those more accurate algorithms is you can use them and expand them into novel multimodal models. So this is the kind of model that's not available without the kind of data that you have access to in certain healthcare settings, because not only do you have images, not only do you have biomarkers, lab tests, you also have other aspects over a course of time, repeated measurements, tabular data, other time series data you need to integrate. And you can then take these novel multimodal algorithms and you can develop novel endpoints. So because you're collecting these images at regular time follow-ups over the course of a clinical trial for late-stage drug development, you can ask yourself, what if we could get something sort of approved and validated, could we use to predict whether or not a treatment was working without having to wait until patients died, without having to wait until they had a clinical progression of their disease? What if we could tell early on from 1 follow-up, 2 follow-ups, 3 follow-ups, say 6 weeks again, and 6 more weeks, whether or not a particular drug was going to work? And if it's not working, you could make the ethical decision to switch them over to a different treatment arm, or perhaps it would decrease the overall cost of doing particular reads. As we know, global drug development is a vastly expensive thing, some drugs taking upwards of $1 billion in order to bring to market. And a lot of that has to do with this careful curation and rigorous use of data. So if we can use that data and bootstrap a model that will allow us to use less data in the future, it's a win for the pharmaceutical industry in terms of being able to bring drugs to market at a cheaper cost. But it's also a win for the patients who get to switch over to better treatments. And eventually, as these models mature, get validated, and progress, we can take them into a medical context where they can be used on a daily basis in order to improve patients' lives or augment clinical decision-making. And we're not there yet, but we're now at the point where we can start using this data and the plethora of off-the-shelf algorithms that are readily available in order to test and validate these hypotheses and get one step closer towards that dream of bridging artificial intelligence and medical decision-making. Now, in the clinical trial context, one thing that we would be sort of loath to not include is this notion of patient stratification. And this is extraordinarily important in terms of reducing the cost of clinical trials, but also improving their chance of success in terms of getting to the right answer. And that's that when you randomize patients across clinical trials, you don't just flip a coin with every patient. You try to balance the patient characteristics. But patient characteristics have to be very carefully defined. You might want patients with similar body mass index to be balanced across the placebo group and the active group, for instance. But you might also want to balance on how long they've been smoking. You might also want to balance on other blood biomarkers they have, or their sex. You might also, in the case of these algorithms that we develop, be able to balance on subtle features that are less interpretable, that only come out after the analysis of millions of labeled images, more images perhaps than any radiologist could see in their own individual lifetime. And these features, so-called radiomics, or in sort of a parlance of genomics, they could allow us to make decisions that go beyond the current realm of individual decision-making in terms of clinicians. So whereas in computer vision applications, for instance, in ImageNet, to classify whether something is a dog or a cat, you're trying to reach human performance, and surpassing it is relatively difficult. In the case of medical imaging, there may be room to go far beyond what a clinician can see because some of these clinical features are extraordinarily subtle. So taking one step forward then, and thinking about what can be true tomorrow, we've talked about augmented decision-making, but the future really is more of a blank slate. We don't know exactly where we could go if we had access to these kind of images. So we don't know if you had the ability to predict how long someone would live, given certain characteristics about them from an imaging perspective, when integrated with other demographic data and biomarker data. We don't know how to build that model yet. But if we could build the model, what would it look like? We don't know what we might be able to use it for. The particular applications are a little bit beyond the scope and somewhat difficult to imagine because it's so far beyond what we've been able to do today. But we can ask, what can be true tomorrow? And what are the basic steps that we would take to get there? And I think as an industry, it's worth taking stock of what the academic and industry partnerships from Facebook AI research, from DeepMind, from OpenAI have to offer in terms of rapidly developing and deploying these solutions. So in terms of timescale, we know that at a slow pace, these use cases aren't going to change. We know that at a leisurely pace, the data is not going to change. We're still going to collect data for clinical trials. We're still going to rigorously curate and manage that. But quickly, algorithms are going to change. It seems like every week, every month, you go to Papers with Code, you see that there's a new state of the art on a variety of benchmarks, whether that's natural language processing with the glue benchmarks or super glue benchmarks using transformer architectures, whether that's the advent of transformer architecture surpassing convolutional neural networks in imaging, or no multimodal combinations of those, perhaps applications to time series. We know that these algorithms are going to change rapidly. So speaking on those algorithms, what we can apply today, because we can't change the data and we can't change the use case yet, what is the progress we can make and where does the pharmaceutical industry fit in? So we know a given, say, trajectory for a medical image might look a little bit like the following. You may have what's called a CT scan, and this is shown as a horizontal slice. You could imagine putting your hand at your belly button and chopping all the way through horizontally. If you could take an image of that slice, that's what you'd see. Now we know that we'd stack these together, but just looking at one, you might notice if you were a radiologist, particular abnormalities. You might categorize them. You might notice that there's some lesions or tumors that you might want to segment. So what if we could do that in an automated or a semi-automated way? So rather than paying someone to do it, to measure it manually, we just had it automatically done. And then given those regions of interest, what if we could generate features? We would call these radiomic features in sort of a shout out to genomics or proteomics. We could generate features that describe this abnormality. And then you can build a classifier on top of those numbers that incorporates other kinds of data. That data could be clinical data. That data could be patient reports. It could be text data. It could be biomarkers, or it could be time series data. What would that look like in a mathematical sense if we were to break that down? When we'd have an image, we turn it into features. That is nothing more than object detection. And object detection in the last 5 years has had remarkable advances. If you just look at the COCO dataset results. Well, that's nothing other than what radiologists do, where they're looking at a series of 2D images going slice by slice, say horizontally through your chest, looking for abnormalities. Let's say you notice 3 lesions or N lesions, and you associated with each of those a feature vector. Doesn't matter yet how you'd get there. Maybe you'd pull the final penultimate layer of a convolutional neural network or a transformer in order to have a feature vector that you'd later use for your classifier. Or perhaps you define it with wavelets or incorporate some other a priori defined measures like volume, surface area, roughness, heterogeneity, things like that. However you generate that, that's nothing else but a list of vectors. And to a machine learning scientist, this is really attractive because now you have numbers that you can work with. And if you have outcomes that derive from your use cases, you can use those in order to make predictions. And when you make those predictions, you can use that as feedback to update those vectors. And those vectors become better and better predictors or representers of the phenomena, the phenotypes that you're interested in a clinical context. What's key here, though, is that these models don't just build themselves. They need some sort of, typically, target in order to train to. Self-supervised learning has provided a dramatic sort of advance in the last, say, 2 to 3 years. But in the clinical context where we have access to data for every patient, we know typically, unless they have been lost to follow-up, how long they survived. And we know how long it took before they had a progression event, depending on the clinical trial. So building models on this, we now have a way to go through the model in order to improve this representation for these lesions. And what's particularly exciting is that these representations, as they get better and better and better with more and more data, become more specific. So going back to the fact that pharmaceutical industry has a large amount of data because global drug development is a data generating and interpreting machine at its core. And going back to the fact that we know that models get better with more data typically, as long as that data is germane to the problem at hand. We know that these features can get better and better and better with every single iteration of the model, with every single prognosis or prediction made. So when we go back then to the question, what can be true tomorrow, given what we know about today? We can look forward to novel endpoints. We can look forward to patient stratification. We can look forward to artificial intelligence in triage cost reduction, where you know that you're not necessarily able to lower the cost or obviate the need for every radiologist, but you may be able to route some radiologist predictions towards an AI. So we know, for instance, that there are, the FDA has approved in terms of lung cancer screening, humans to be obviated by a second reader from an AI. And we know that in the pharmaceutical industry, perhaps, we can start to think about how we would use that, how we would apply that. But it's also important to remember that at the end of the day, what can be true tomorrow always comes back to a better model. And then that model can be used to apply to any other endpoints, the kinds of endpoints that we can't even yet imagine. Thank you.
Wow, awesome job, Kevin. Thank you so much for sharing with us. If you could stop sharing your screen, I know the virtual audience wants to give you a huge virtual round of applause. Thank you so much for sharing with us today. For the audience, it's time for you to make your way to your next session. Along the way, make sure you accept your connection request and take some time to check out our amazing exhibits. Thanks so much, and we'll see you around.